Hello, welcome to Learning Theatre. In today's video, we're going to talk about questions and answers from glycolysis. In our previous video, we did a detailed lecture on regulation of glycolysis. And so today, we're going to talk about some questions and answers. We're going to answer some questions under glycolysis. So the first question we have here is, what is glycolysis? What is glycolysis? When we say glycolysis, this is a central metabolic pathway that breaks down glucose into pyruvate, producing energy in the form of ATP and then reducing power in the form of NADH. So let me write that down. So glycolysis is a central metabolic pathway. Glycolysis is a central metabolic pathway that breaks down glucose that breaks down down glucose glucose into pyruvate producing energy producing energy in the form of ATP in the form of ATP and reducing power in the form of NADH in the form of NADH. Yes, so glycolysis is a central metabolic pathway that breaks down glucose into pyruvate, producing energy in the form of ATP and reducing power in the form of NADH. And this consists of a series of enzymatic reactions that occurs in the cytoplasm of a cell. And then it's also a process divided into two phases. We have the energy investment phase and then the energy payoff phase. So this is what is known as glycolysis. When we break down glucose, producing two pyruvate molecules, and then in the form producing energy in the form of ATP and also in reducing power in the form of NADH. And then the second question we have is, where does glycolysis occur? Where does glycolysis occur? So glycolysis occurs in the cytoplasm of a cell. So glycolysis occurs in the cytoplasm of a cell. Yes, so the process of glycolysis occurs in the cytoplasm of a cell, and this location is very important as it allows for rapid glucose metabolism without the need for oxygen. And then this makes glycolysis the fundamental process in both aerobic and anaerobic conditions. Yes, so glycolysis occurs in the cytoplasm of a cell. The third question we have here is, what is the starting molecule of glycolysis? What is the starting molecule of glycolysis? And the starting molecule of glycolysis, or the starting substrate, is what is known as glucose. So glucose is the starting substrate or starting molecule of glycolysis. So glucose, glucose Glucose is a starting molecule or substrate of glycolysis. So this glucose is a cis carbon sugar, and then it is phosphorylated by by a hexokinase enzyme to form or to convert it to glucose 6 phosphate, and then initiating the pathway and then trapping glucose within the cell. And the next question we have here is, how many ATP molecules are consumed in energy investment phase of glycolysis? How many ATP molecules are consumed in the energy investment phase of glycolysis? Yeah. So we have two molecules of ATPs that are consumed during the energy investment phase of glycolysis. So the number of ATP molecules that are consumed in the energy investment phase of glycolysis are two molecules of ATP. So two molecules of ATP. Yes, so in the energy investment phase, two molecules of ATP are consumed, and then this energy are used to phosphorylate glucose, making it more reactive and then facilitating its breakdown. So these two molecules are used to phosphorylate glucose in the energy investment phase. And then the fifth question is saying, what are the end product of glycolysis? What are the end product of what are the end product of glycolysis? What are the end products of glycolysis? So the end product of glycolysis, we have two molecules of pyruvate, two molecules of ATP, and then two molecules of NADH. So let me do that here. So we have two molecules of pyruvate, two molecules of ATP, and two molecules of NADH. Yes, so the end product of glycolysis are two molecules of pyruvate, two molecules of ATP, and also two molecules of NADH. Yes, so these are the um, end product of glycolysis. And the question 6 is saying that what is the net gain of ATP in glycolysis? What is the net gain of ATP in glycolysis? So the net gain of ATP in glycolysis are two molecules of ATP. The net gain of ATP in glycolysis are two. Remember, we will produce two and four ATPs, and then we use two ATPs, and then the remaining two ATPs. So the net gain are two molecules of ATP. So let me do that here. Two molecules of ATP. Yeah. So the net gain of ATP in glycolysis are two molecules of ATP. So while four ATP while four ATP molecules were produced, two ATP molecules were consumed during the energy investment phase, which result in a net gain of two ATP molecules. Yes. And the seventh question here is, what coenzyme is reducing during glycolysis? What coenzyme is reducing during glycolysis? The coenzyme that is reduced um, during glycolysis is what is known as nicotinamide adenine dinucleotide. Nicotinamide adenine dinucleotide, that is NAD+, which act as a coenzyme and then it is reduced to NADH during glycolysis. So let me write that down. So nicotinamide adenine dinucleotide, that is NAD plus, which is reduced to NADH during glycolysis. So during glycolysis, yes, yeah. So the coenzyme that is reducing during glycolysis, what is known as nicotinamide adenine dinucleotide, NAD plus, which is reduced to NADH during glycolysis. The eighth question we have is that what is the enzyme that catalyzes the first step in glycolysis? Yeah, so the enzyme that catalyzes the first step in glycolysis is what is known as the hexokinase. Hexokinase. This is the enzyme that catalyzes the first step of glycolysis. So hexokinase. Hexokinase. Yes. So this enzyme phosphorylates glucose to form glucose 6-phosphate, and this requires the input of one ATP molecule. Now, the next question. In which step of glycolysis is ATP directly produced? In which step of glycolysis is ATP directly produced? So, the step of glycolysis in which ATP is directly produced is during the substrate level phosphorylation step. That's the substrate level phosphorylation step of glycolysis. That is when ATP is directly produced. So let me write that down. So ATP is directly produced in the substrate level phosphorylation step of glycolysis. Yes, so this is the step in which ATP is directly produced, and this step is specifically during the conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate. Yes. And the next question is also saying that what is the rate limiting enzyme of glycolysis? What is the rate limiting enzyme of glycolysis? And the rate limiting enzyme of glycolysis is known as phosphofructose kinase, um, phosphofructose kinase enzyme. Yes, so this is the rate limiting enzyme in glycolysis, phosphofructose kinase. So phosphofructose kinase. Yes, so this is a um, rate limiting enzyme of glycolysis, and this enzyme catalyzes the phosphorylation of fructose 6-phosphate to fructose 1,6-bisphosphate, and is a key regulation step that governs the overall process of glycolysis. Yes, so it involves the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. Question 11. What is the significance what is the significance of the rate limiting step? The significance of the rate limiting step. The significance of the rate limiting step is control. The rate limiting step here is controlled by phosphofructokinase, which serves as the critical checkpoint in regulation of glycolysis. So this rate limiting step is controlled by phosphofructose kinase, and it also serves as a critical checkpoint in regulation of glycolysis. So let me write that down. So the rate limiting step, the rate limiting step is controlled by phosphofructose kinase, and it serves as a critical checkpoint in regulation of glycolysis. Yes. So the rate limiting step serves as a critical checkpoint in the regulation of glycolysis, and it responds to cellular energy needs, ensuring that glycolytic pathway matches the demand for ATP. Yeah, so this is the importance of the rate limiting step. And question 12. What is the purpose of the energy investment phase? What is the purpose of the energy investment phase? The energy investment phase serves to activate and then prepare the glucose for subsequent reactions. It serves to activate and prepare glucose for subsequent reactions. So the energy investment phase, this serves to activate glucose and prepare glucose for subsequent reactions. Yes. So this is the purpose of the energy investment phase. It is there to activate and prepare glucose for subsequent reactions. Yes. And this energy investment phase also involves the phosphorylation step that make glucose more reactive and then facilitate its subsequent breakdown. Yes, so the energy investment phase also serves as a phosphorylation step that make glucose more reactive and then facilitate its subsequent breakdown. Now, question 13. How is ATP generated during glycolysis? How is ATP generated during glycolysis? ATP is generated through substrate level phosphorylation during glycolysis. ATP is generated through substrate level phosphorylation during glycolysis. So let me write that down. ATP is generated during substrate level phosphorylation phosphorylation during glycolysis. Yeah, so this is how ATP is generated, and this occurs when high energy phosphate groups are transferred directly to ADP to form ATP, and then catalyzing and contributing to the cell um, energy currency. Yeah, so when it does that, we generate ATP in the process. Number 14. What is the fate of pyruvate under aerobic conditions? What is the fate of pyruvate under aerobic conditions? Under aerobic conditions, pyruvate generated in glycolysis enters the mitochondria and undergoes further oxidative metabolism in the citric acid cycle. Yes, so during aerobic conditions, pyruvate that is generated in glycolysis will enter the mitochondrion and undergoes further oxidative metabolism in the citric acid cycle. Yes, well, let me write that down. So, under aerobic condition, aerobic aerobic condition, pyruvate that is generated enters the mitochondrion mitochondria and further oxidative undergoes further oxidative metabolism oxidative metabolism in the citric acid cycle. Yes. So under aerobic condition, pyruvate, which has been generated during the glycolysis, enters into the mitochondria and undergoes further oxidative metabolism in the citric acid cycle. And this process ultimately leads to the production of more ATPs through oxidative phosphorylation. Um, Prince, thank you for watching. Please like this video, comment in the comment section, and also share this video to your colleagues. And don't forget to subscribe and then press the notification bell. We will bring you part 2 of these questions and answers under glycolysis. See you in our next video.